immune system optimization in a variable world. And some of the recent and in many cases unpublished work coming out of my lab right now. So I want to first start out by saying that parasitism is not a new invention, and pose the question that really, if hosts have been selected by the negative consequences of parasites for so long, why do we still get sick? Oops. There it goes. Okay. So one of the explanations is that parasites can evolve to evade host defenses, right? We're evolving, but the parasites can evolve just as quickly. This is, for example, a phylogeny from the H3N2 influenza A strain of the virus. And you can see from the phylogeny that most lineages do not persist for very long. And that's because the immune system is really driving the evolution and antigenic drift very strongly of this virus, so that it has to constantly adapt in order to continue infecting humans. But even if you do recognize a threat, an optimal immune response is not always a maximal one. And I love to give this example of this squirrel with a flamethrower when I teach immunology to my students, for example, just to highlight the fact that the immune system is very powerful. It's really good at killing microbes, but there's a lot of collateral damage that can happen if you're not careful about controlling the strength and magnitude of immune responses. The immune system is pretty well organized, but there are some problems that could arise from insufficient negative feedback or from immune responses that are too robust. So you don't necessarily want to give a squirrel a flamethrower. And by the same token, you want to be able to control the magnitude of your immune responses. Immunity is costly and constrained in many other ways as well. So it takes energy and resources in terms of, for example, you can think of a hamburger or, whatever your favorite food is. You have to acquire a lot of that energy in order to fuel an immune response. Parasites, if you're infected, are trying to suck away your energy to exploit it for their own gain. And so energy can be a really important limiting factor in driving the magnitude of an immune response. Immunopathology, that squirrel with a flamethrower, for example, can be thought of as a double-edged sword, right? You're really good at killing microbes, but you can also cause substantial damage to the host. And then there can be genetic or pleiotropic costs to an immune response. That is, fundamentally, if you're using a pathway or you're polarizing a T cell response toward the production of one arm of the immune response, that could come at the cost of other things that are being regulated by those pathway components or other immune responses that are relying on things that are being negatively regulated during an immune response. And finally, we have multiple fronts. And this is the idea that it can be really hard to simultaneously optimize an immune system when you're dealing with a real diversity of threats that require different types of defenses. So I like to use this example of a rabbit, for example. It has two predators here, a hawk and a snake. It could, for example, evolve bigger legs where it could rise above the grass and jump faster than the snake, but that might make it more susceptible to being seen by the hawk, right? And so there's this idea of this fundamental trade-off. And this is certainly true for immune systems. So thinking about our T cell polarization, for example, if you're polarizing toward a type 1, Th1 response to respond to an intracellular microbe like a virus or, a coronavirus or something like that, it could come at the cost of Th2 mediated responses, the type 2 responses that might make you better able to defend yourself against worms or something like that. As a result of all these costs and constraints, hosts have evolved complex and tightly regulated immune systems. There's a lot of negative feedbacks here. There's a lot of checkpoints to make sure that things don't get out of control. Here, for example, is some of the major arms of the fly immune response. So this is pretty standard across many other insect species as well, where we have our pathogens down here. They're recognized in a variety of ways, which can lead to the production of a melanotic nodule. This is kind of a sticky black substance that can surround parasitoid eggs, for example, or nodulate bacteria together so that they can't spread throughout the insect hemocell, the body of the insect. They have cellular immunity, so they can go around with their phagocytes and they can snap up roving bacteria or whatever else is infecting the host, and they can digest those bacteria just as a monocyte or a macrophage or a neutrophil would. And these hemocytes have many other roles as well. They're involved in development, for example, and they're involved in encapsulating foreign objects. For example, if a parasitoid egg gets into the body, your cellular immune response is going to be really important for that. And then we have our signaling cascades, our signaling pathways. And these are really important for producing things like antimicrobial peptides, which are going to punch holes in the cell membranes of bacteria. They're going to produce other effectors like transferrins, which can take iron away from microbial availability so the microbes can't exploit it. This is a way of nutritionally restricting microbial growth. So there's many different ways in which an insect can control a foreign invader. But there's also an important point here, and that's that these immune systems aren't evolving under constant parasite pressure. There's a lot of feedbacks that are coming in from the ecological side of things. For example, you can think of a population of any host that you want to think of, and it's infected with a parasite, right? And so as the incidence of infection goes up, and maybe this parasite is pretty good at killing hosts or something, the strength of selection on a gene that provides resistance to an individual against this parasite is going to increase in frequency in the population from 0 to something that's not 0, right? But as that resistance allele increases in the population, those resistant individuals aren't going to get infected anymore. And if they're not infected, they can't spread the disease to others, in which case that infection prevalence is going to go down, which is going to reduce the selective pressure on that resistance allele. And so in many cases, it can be really hard to have a resistance gene fix in a population because of this varying selective pressure from the epidemiological side of the parasite transmission dynamics. And if we zoom out to thinking about infections as they work across different biological levels of organization, from the molecular to the organismal to their impact on host populations and communities, things can get pretty complicated. So natural immunological variation, for example, among different host species, or even among different individuals of the same species. For example, immunological variation between tick larvae and tick adults, for example, can drive differences in the overall transmission and incidence and spillover of an infectious disease like Lyme disease. I just like to give this example because I think it helps illustrate that when you have, for example, many different hosts down here, we have a white-footed mouse and we have a squirrel and we have a lizard. White-footed mouse is really great. It's very, what we call competent for the disease. It can have very high Borrelia titers. And if a tick bites it, there's a very high probability that those bacteria get transmitted. A lizard, on the other hand, may not be very competent. So it's more resistant, for example, to the infection, and so it's less likely to drive transmission. And so when we think about immunological variation at this level, we really have to think, and if we want to ask the question, what's our risk of getting Borrelia? What's our risk of getting Lyme disease? It requires knowledge of this entire the entire dynamics of this whole system. And so my lab, we like to think about these different interactions at these different biological levels of organization, from the molecular to the organismal, to thinking about dynamics and how these kind of complex ecological and evolutionary feedbacks drive patterns of disease in populations and communities. So today I want to ask a question. How does host ecology shape the evolution of innate immune systems in insects? And I'm going to be talking about 3 different research umbrellas that we have going on right now. The first part will be thinking about developmental constraints on immune system evolution, thinking about those, for example, those ticks where you have larvae and adults that have variation in their immune responses, and how that drives the evolution of immune systems. Then we're going to be thinking about the ecological consequences of defending multiple fronts. If you're being infected, if you're that rabbit, right, and you have your predator, your 2 predators, that's a multiple fronts issue. But by the same token, a host that's facing multiple parasites could have a multiple fronts issue. And so we're going to be looking first at the ecological consequences of defending multiple fronts. And then we're going to be thinking about the evolutionary consequences of defending multiple fronts. Okay, so I want to pose a question to you, and that's the problem of maintaining an immune system across ontogeny. Ontogeny being the progression of an individual from birth to old age, all right? So for example, we can think of my flower beetles. Flower beetles are a stored grain pest. They are found throughout the world, and they are holometabolists. They're a beetle, and so they have a larval stage, then they go into metamorphosis, into their pupal stage, and then they emerge as adults. And they live mainly in grain elevators and feed mills and anywhere where humans store their grain. But they live, for example, in different areas of a flower bin. So if you go out to a grain silo and you sample the top of that grain silo, you're going to find a lot of adults and pupae. If you delve farther down into that grain silo, where it could be warmer, could be more humid, you could have older material, higher density of individuals, you're going to find the larvae down there. So even though ostensibly these individuals have the same or similar ecological niche, when you get down far enough, you see that there's differences in their niches and potential exposure to microbes. And I think this is recapitulated nicely when we think about something with an even more drastic ecological shift during ontogeny. So mosquito, right, the adult stage is terrestrial for the most part. It goes around, the females particularly, trying to find a blood meal. They're exposed to microbes in that blood meal. They're exposed to microbes in the nectar of the flowers that they're drinking to get liquid from. But they lay their eggs in the water. And the larvae are swimming around and they're being exposed to aquatic microbes and potential aquatic pathogens. Same thing with the pupae, right? So here we have very different ecological niches where we could have a very different milieu of pathogens and parasites that the different life stages are being exposed to. So for example, in our flower beetles, we did this experiment. We wanted to know, does exposure to a microbe in the larval stage in the early life influence the immune responses or immune system maturation in subsequent stages? And to investigate this question, we had our larval Tribolium castaneum beetles here, and we had this parasite. And this is a gregarine parasite. It's a protozoan parasite. These types of parasites are actually really ubiquitous in insects. If you work on mosquitoes or you work on, maybe Drosophila has them, I don't remember, but if you work on Orthoptera, so if you're a cricket person, locust person, or most beetles, if you just go out to your lab colonies or you go out to the wild and you pull the guts out of your insects, you're likely to find these parasites. So in our flower beetles in particular, when we sample in the wild, about 50% of our individuals we find have parasites that are these protozoan parasites. Different species, but overall they're a pretty ubiquitous potential selective pressure on the host in the wild. And so what we did is that we infected our larvae with these parasites. Now they're gut parasites, and so when the host goes through metamorphosis, it sheds its gut lining, and it sheds the parasites at the same time. So the adults emerge uninfected, just naturally. We don't have to treat them with anything. And so we infected these larvae. We measured their gene expression of many different genes, but I'm showing you two here. This is cecropin-3, which is an antimicrobial peptide, and PGRPLC, which is involved in peptidoglycan recognition. These are two genes that we know, for example, are really important for antimicrobial responses in the beetles, antibacterial responses. And you can see, first of all, that these are in the guts. Individual larvae who are infected with these gregarines have significantly lower expression of both of these genes if they have gregarine infection. Now, once they shed their parasites, you can see in the pupae, there's not much difference in either stage, although there's still maybe a little bit of a trend towards lower expression. And that could just because these pupae actually don't have much of a gut at all. They have a tiny little segment with a bunch of stem cells that are going to repopulate the gut in adulthood. But then when they emerge as adults, you can see that there is still, and this is particularly in the case of PGRPLC, this is significant. They have lower expression, even though they're not infected anymore. They have lower expression of these genes that persist into adulthood. And we think more broadly about immune system regulation in different life stages. We can see, for example, that if we look at PGRP and cecropin-3, and we look at their co-regulation, so how is the host managing the expression of a module of immune genes that might be together really important for an effective immune response. We can see that this too differs between different life stages. So in general, particularly in the stages that are moving around, there's a really tight co-expression pattern between this recognition protein and this antimicrobial peptide. And you can see that here in the expression pattern. But you can see also that larvae overall have lower expression of both of these genes. And adults have higher expression of both of these genes. Pupae, meanwhile, this tight relationship seems to be disrupted in the pupae. So the regulatory machinery that's responsible for the co-regulation of these genes seems to be broken down in this particular life stage. And this leads to the question, there's this hypothesis out there, this idea of adaptive decoupling. The idea that if you, one of the big advantages of metamorphosis in insects is that you can tailor your immune responses to the particular ecological niche that you are living in during your different stages because of this ability to remodel your body, but also change the co-regulatory dynamics of immune gene modules, for example. And so we can see that there's something going on here, right? There's some decoupling going on here, particularly in the pupal stage. But is this an adaptive decoupling issue, or could it be a constraint? Is it actually adaptive at all? And so I wanted to think about the maturation of immune responses in insects as they go through development in order to think harder about this problem of trying to figure out if something's adaptive or actually reflecting a constraint. And I want to point out this example of Drosophila melanogaster hematopoietic organ maturation. So the hematopoietic organ is where hemocytes are made, those phagocytic cells that are going around and either chomping up microbes or encapsulating something like a parasitoid egg here. So this has been melanized and encapsulated by hemocytes. And as a third instar larva, these guys can be champs at this. So they're able to mount a pretty quick response against a given parasitoid egg. But if they're infected earlier, so in the L1 or L2 stages and earlier instars, they don't stand much of a chance against these parasitoid eggs. And the reason is the hematopoietic organ doesn't really fully mature until the third instar. And so this is a developmental constraint on the use of your cellular immunity to defend yourself. Just based on the developmental aspects of these individuals. So they can still get infected, they're still moving around and eating, but they're not well defended because of this developmental constraint. Fetal oxidase is another example of this, is that in part responsible for that black sticky substance around the egg. And it turns out it oscillates from high to low during molting between each larval stage. And the reason is that PO is a pleiotropic molecule. It's involved in multiple functional activities inside the host. So for example, it's involved in parasite melanization, like we see here, but it's also involved in tanning the cuticle after every molt. So the larva has to shed its exoskeleton so that it can grow bigger, and then it builds it again. And that molecule is helping to tan the cuticle. And so what we see with fetal oxidase availability is that because of this cuticle tanning, the levels oscillate during every molt, but transiently, when they're being used to tan the cuticle, they can't also be used to tan or to provide melanization responses against parasites. And so a study, for example, McNeil et al. In 2010 found that these oscillations correlate with the ability of these individuals to survive fungal infection, suggesting that development can drive transient increases in susceptibility, this developmental pleiotropy. And so when we think about these different mechanisms, these, the hematopoietic organ maturation and these phenol oxidase pleiotropy issues, I want to ask the question, how pervasive is developmental and immunological pleiotropy anyway? And so we've already seen that the phenol oxidase is involved in cuticle melanization. It's also actually involved in neurotransmitter production. It helps produce dopamine, for example. And it has these immunological functions. So it's clearly pleiotropic, at least some portions of these pathways, some portion of the pathway is. Toll pathway is famous for being pleiotropic. So it's actually won 2 Nobel Prizes, components of the toll pathway. The first one being for its role in developmental patterning and dorsoventral patterning. It's also involved in hematopoiesis, and it influences insulin signaling and metabolism. But it's a fundamental signaling pathway for the production of antimicrobial peptides during the immune response and for hemocyte activation. And so once again, at least some components of the toll pathway are incredibly pleiotropic. And we can do this again and again for different canonical immune pathways that we know about. We can point to molecules within those pathways that are clearly pleiotropic between development and immunity. So the answer here is that at least what we think about in terms of the canonical immune responses, pleiotropy is nearly ubiquitous. And so we started thinking, well, if pleiotropy can inhibit or modulate the maturation and susceptibility of insects over development, right, in different developmental stages, could it also constrain their evolution? And the reason we wanted to ask this is because developmental genes are kind of famous for being under purifying selection, right? if you mess with a developmental gene, particularly one that is kind of in a tight module with a bunch of other developmental genes, you might develop like a seventh leg, or something could go terribly wrong. And so you don't really mess with that machinery. Evolution is really good at kind of paring down deleterious mutations in those genes. Immune systems, on the other hand, are kind of famous for evolving under adaptive evolution, right? They're undergoing positive selection in many cases. A lot of the signatures of positive selection that we see in any organism that you pick, right, whether it be humans or apes or mice or whatever else, immune genes tend to fall out in that list. They tend to be, at least some of them tend to be under positive selection. So this extensive pleiotropy between immunity and development could be somewhat constrained or in conflict in terms of how evolution might act on this. So we wanted to actually go and look and ask, are immune genes that are also pleiotropic under stronger purifying selection than immune genes that aren't pleiotropic? And to do this, my grad student, Tin Yo, turned to the Drosophila melanogaster subgroup here. There's sequenced genomes for each of these species on this phylogeny. And there's within the subgroup, there's also some mini genomes that you can use, for example, population genetics and things like that. So she turned to the Drosophila melanogaster subgroup, and she wanted to ask how many genes are pleiotropic between immunity and development. And so she went to she conglomerated a bunch of different data sets. She had transcriptomics data, she had Go analyses Go terms from previous analyses, most of which were manually annotated, and she used fly-based annotations and things like that to come up with the percent of immune genes that were potentially pleiotropic. And so here we have genes that are known to be involved in the immune response in general. They're annotated as defense response, whatever. We have genes that are involved in development over here. We have some that are involved in immune development. We weren't quite sure how to categorize these, things like the development of hemocytes, for example. And then we have those that are just straight up pleiotropic. Okay, so these are genes that are involved in the immune response, but also involved in development. And when we look at how these are distributed, so we have recognition genes. These would be things like our PGRPs. We have signaling genes. These are those transducers in the toll pathway, for example. Effector genes are antimicrobial peptides and such, antiviral genes, and then other types of genes. And what we see, first of all, when we just look at the distribution, is that pleiotropy is really concentrated in the signaling genes. There are a few in the recognition, effector, and antiviral responses, but signaling genes tend to be pretty pleiotropic. And when we actually looked at the signatures of selection on these genes, so first I'll draw your attention to this plot, this violin plot over here, where we have the DNDS over the entire melanogaster subgroup, so those 5 species. And we have non-pleiotropic immune genes here. We have pleiotropic genes that are both pleiotropic for development and immunity here. And then we just have your non-pleiotropic developmental genes. We can see, first of all, that developmental genes have the lowest DNDS. So the lower your DNDS, the stronger purifying selection, for example. So they certainly seem to be under purifying selection, although not all of them are. Right? Our immune genes, the non-pleiotropic ones, have significantly higher DNDS, which means that they're under less purifying selection and potentially under stronger positive selection, depending on how you want to interpret that. And then our pleiotropic are somewhere in the middle. So they're actually significant. The DNDS is actually significantly lower than the non-pleiotropic genes, but it's not a huge effect size, right? So there's something there, but there's a lot of variance to explain. And so we turn to the actual pathways that we're interested in. For example, the IMD signaling pathway, the toll pathway. We've already posited that the toll pathway is very pleiotropic. And we basically took the gene or the genes that are involved in these different pathways, and we tried to visualize what their pleiotropic status. And so you can see, for example, that in the toll pathway, we have some components here that are pleiotropic between development and immunity, MyD88, for example, and it has an intermediate DNDS level. So it's certainly not under positive selection, but then it's associated with a pleiotropic gene that does have a higher DNDS. And then Conversely, we have dorsal here, which is incredibly under purifying selection, cactus, which seems to be as well. So you can see that even within these pathways, there's a lot of pleiotropy in these signaling pathways, but the distribution of DNDS, there's not really a clear pattern emerging. Over here in the IMD pathway, we can see that many of our pleiotropic genes are actually associated with interacting with non-pleiotropic immune genes, which potentially suggests that at least some of these pathways, pleiotropy could be modulated by adapter proteins or partners that can help modify the pleiotropic costs and constraints of the use of that protein. So to conclude this first part, I just want to point out that complete metamorphosis in insects, whether it be your Hymenoptera, your bees and things, your beetles, your flies, or your moths, complete metamorphosis may allow decoupling of stage-specific immune modules. But pleiotropy can interfere with immunological maturation. And in terms of developmental pleiotropy and how it can constrain immune responses evolutionarily, we don't really have a clear answer. But we're still, it seems like it at least weakly affects positive selection. But we're still looking into things like ortholog bias. One of the things that you can do to relieve pleiotropy potentially is to duplicate a gene and diversify it. So we're thinking about that. There could be compensatory mutations. So it could be that overall a particular sequence has a low DNDS or a high DNDS signature. But if you look at the particular protein domains within those proteins, you could have regions that are under positive selection and regions that are under purifying selection that could somehow cancel each other out, for example, or modify the structure of the protein in certain ways. And then we're interested in extending this beyond Drosophila to see if the patterns that we're seeing in Drosophila are generalizable, or if they're kind of a quirk of the fruit fly, which we know is wonderful model in many cases, but isn't always strictly generalizable to insects. These concepts apply to developmental dynamics in many other organisms. So I like to think about plant development because there's a lot of pleiotropy in plant genetics in terms of tension between development and immunity across its ontogeny. But humans at the same time, right? So as we progress from birth to old age, our immune systems are really dynamic. They mature. There's some passive immunity at the beginning of our lives. Our T cells and B cells mature, but maybe at different rates. We tend to be biased towards Th1 versus Th2 at different points in our lives, which can influence our susceptibility. During pregnancy, right, we have these transient tolerance responses to make sure that we don't reject a fetus that's expressing, paternal antigens, which can really influence immune responses. And as you age, your immune system starts to decline, but not homogenously. So some arms the immune response, and this is becoming very apparent in the response to coronaviruses, for example, right? Some of those Th1 responses in particular decline more rapidly than things like the Th2 responses. Okay, so that was development. Now I'm going to shift to talk about the ecological consequences of defending multiple fronts. I want to start by talking about flower beetles again. So if you guys have ever picked up a population biology textbook or a theoretical ecology textbook, this is literally a textbook example of apparent competition. The idea that two host species that are in the same environment, in the same niche, are competing with each other. They're competing for each other with resources, but sometimes external factors, for example, a parasite, can modify the outcome of competition. And so in this case, these are experiments done back in the 40s and 50s that have held true in terms of their illustrative value. We have two beetle species, Tribolium castaneum and Tribolium confusum. So that's T and F here, castaneum, confusum. In the absence of any parasites, castaneum outcompetes confusum. So this is the mean population density here. This is time. And you can see that confusum, when they're put in the same arena, confusum will decline to nothing and castaneum will completely exclude them. All right. However, if you add this parasite, this is Adelina tribolii, it's a protozoan parasite, and you throw it into the mix, suddenly confusum does pretty well, whereas castaneum just bottoms out and can even be excluded from the population by confusum. So this parasite is mediating what we call apparent competition between these two species, and it can flip the outcome of infection. And so in modern times, in my lab, we have these two species in my lab. And we've started to try to recreate these competitive dynamics in order to play around with things like environmental variables or co-infection. What we find, for example, is that when we have our two beetle species and we infect them with this bacterium, Bacillus thuringiensis, which in many ways has very similar life history characteristics to the protozoan parasite that I showed you before, we see once again, this is hours post-BT infection in the proportion alive, that our confusum beetles are much better able to survive infection than our Castanian beetles. And we've actually repeated this for several different wild-caught populations. And overall, because confusum is just much better able to survive infection. And it seems to be mostly mediated by resistance. So they're better able to kill the bacteria. But flower beetles have to deal with multiple fronts all the time. So it's not just, BT is a little bit artificial in terms of, us infecting them in the lab. They do have natural strains of BT that we've isolated from the wild, but they also have all these other parasites, right? I already told you about the protozoan parasites that they get in their guts. There's several species of those. They have other obligate killer parasites like these protozoan parasites that live in their fat body that basically reproduce and eat up all the host fat stores until the host dies. We have this one parasite that we found that seems to have a mix of transmission modes. So it lives in the fat body, but it can also live in the seminal vesicles of the males, where it can actually, it seems like it eats sperm or something, but it can leak out into the environment from those males, or it can even potentially be sexually transmitted or vertically transmitted. So that's a pretty cool parasite. They have Wolbachia, which, if you guys know, is a pretty famous symbiont in terms of having some pretty tricky reproductive modulations of its host. But particularly in confusum, it actually seems to have some benefits in terms of helping produce metabolites and things. And then, really significantly for our beetles, pesticides. Pesticides are an environmental obstacle that they have to face that has certainly exerted a very strong effect on signatures of selection in their genome, for example. And we have a project about pesticide evolution in my lab. I won't talk about it today, but I'd be happy to tell you more about it if you're interested. Okay, so co-infection with multiple parasites could alter the outcome of infection with a focal parasite through several different types of interactions. So for example, if we have parasite 1, BT, for example, and we have parasite 2, these could be our gregarine, those gut parasites, they could compete for resources, right? They could be trying to steal each other's resources. But they could also, they could be stealing resources meant for immunity, right? If the parasites are too good at exploiting this compartment, immunity won't have enough resources to mount an effective response. But the parasites could also be either competing or facilitating each other through immunity as well. So, for example, we saw that our gregarines are down-regulating various immune genes, which could potentially facilitate BT coming along. On the other hand, if one of these parasites upregulates an immune response that's cross-protective, then the immune response can modulate apparent competition between two parasites. So we see that we have apparent competition within the host, modulated by the immune system. And then we have this idea of apparent competition in host communities being modulated by the parasite. And so one of the things that we're really interested in is trying to understand how at these different ecological levels of organization, within the host versus in a host community, how does apparent competition structure competition outcomes? And to start to answer this question, first on the experiment, I'll tell you about the experimental side of things very briefly, and then I'll tell you about mathematical modeling that we've been doing to try to disambiguate some of these things. My postdoc, Nora Schultz, she and my RA2 Sitchin took our two species. We have Tribolium confusum and Tribolium castanium. Remember, confusum is more resistant to BT, and castanium is less resistant. And they took the larvae and they gave them gregarines. Now, in our beetles, it turns out that gregarines are really species specific, sometimes even stage specific. So we haven't yet, despite having 4 or 5 different gregarine species in our lab, we haven't yet found one that infects both of these beetles. But they do affect the beetles in similar ways in terms of they're not very virulent. If you have a lot of them, they might modulate your development a little bit, but otherwise it's a pretty benign infection. But they do have in common that they're incredibly ubiquitous in nature. So if we go out and we infect our beetles, most of them will get infected with these parasites. Then we took these larvae that were either co-infected or not, and we gave them an LD50 dose of BT, and we measured many things, but today I'm just going to show you survival. So it turns out that in the presence or absence of Gregorine infection, the two beetle species had basically opposite effects in terms of the effect of Gregorines on Bt infection. So Tribolium confusum, here we have the solid line is without Gregorines and the dashed line is with Gregorines. Confusum beetles that were infected with Gregorines did worse when they were then infected with Bt than the non-Gregorine individuals did. For Castanium, on the other hand, we actually saw the opposite effect. So Castanium beetles, which normally do worse against Bt, actually did better if they had Gregorines. So here we see that Gregorines are, co-infection is making things worse for Confusum, but better for Castanium. And so we wanted to ask then, to what extent can parasite interactions reinforce or reverse these infection-mediated apparent competition dynamics? And so to look at this, we had this is our initial observation from the 1940s experiments, right, where in the presence of a single infection, we can reverse the outcome of competition. But then if we had co-infection, we could potentially get three different outcomes. Co-infection could reverse that apparent competition effect of the single parasite. It could reinforce it, it can make it stronger, or it could promote coexistence, whereas normally one host species would exclude the other. Perhaps co-infection could make them coexist together. Okay, and so to explore these possibilities, my undergraduate student Faith Rovenholt built a mathematical model where we have, it's based on kind of an SI model where we have susceptible individuals, and then we have individuals of different infection classes. So we have two species here, two host species. Now, individuals could be infected either with Bt or Gregorines, or they could be co-infected. Now, all of these parasites have an environmental stage, which is modeled here, where they can be taken up. So transmission happens as a result of this environmental stage. And the Gregorines are relatively benign infections. They have very few effects in single infections, whereas Bt is an obligate killer. It has to kill the host to be transmitted. And it's pretty virulent in terms of causing mortality in the model. And so in terms of single infection, we can compare the relative recovery and mortality rates among species to look at that initial apparent competition effect mediated by one infection. And then we can layer co-infection onto this. And we can say, what happens when co-infection, what happens when those Gregorines, for example, influence the mortality and recovery rates of each of our host species to that primary infection, to Bt. So that we see in the absence of infection. So this is just, is our model working? This is a good gut check in terms of thinking about how our system is working. We see that in the absence of infection, reproductive rate, so the ability of Castanium to pump out babies versus Confusum to pump out babies, is on this axis here. And then their interspecific competition. So in beetles, they tend to eat each other's eggs pretty voraciously. They're kind of cannibalistic. And Castanium's a little bit better at eating eggs than Confusum. And so when we look at where the actual experimental parameters are just from flower beetle population dynamics, we can kind of put an X marks the spot on our parameter values here. And this is where we're starting our simulations. So in the absence of any infection, Tribolium Castanium is going to win by virtue of the fact, basically, that it has a higher reproductive rate. Okay, but let's throw Bt into the mix. And so if we start out at this white X here, and we throw Bt in the mix, we can say, given that Castanium or Confusum is more resistant or maybe less likely to die from infection than Castanium, how resistant or tolerant, in terms of mortality, does Confusum have to be in order to actually outcompete Confusum to get this apparent competition effect? And so up here you can see that if Confusum has a lower mortality rate than Castanium, it can actually come to dominate the population in the presence of this parasite. And so based on those experimental results from the 1940s, we know that the relative mortality rate, is somewhere up here, and it's similar to the relative mortality rate that we see with Bt. Okay, now we've looked at single infection. What happens if we're thinking about co-infection now? If a co-infection modulates the recovery or the relative mortality rates of each host species to the single infection, what are our potential outcomes? And we can see then that depending, so this is Confusum's tolerance, so it's a relative mortality rate relative to single infection. If you're over here, it means that they're less likely to die if they're co-infected. And over here, it means that they're more likely to die if they're co-infected relative to single infection. And here we have Castanium. So down here, they're less likely to die. And up here, sorry, down here that they are more likely to die, and up here they're less likely to die because it's 1 minus the mortality rate. And so you can see, first of all, that a range of outcomes are possible if co-infection modulates mortality rates. If individuals are, if Confucium is more tolerant of infection, they can come to dominate the population. But there's this big region of coexistence here. So it's possible that co-infection, depending on how it affects mortality rates, it can modulate the coexistence of these species in our population. However, based on our preliminary mortality rate results, we predict that our populations in these particular Gregarine and Bt combinations are probably somewhere here, suggesting that this co-infection is probably going to reverse the outcome of that apparent competition effect modulated by the single infection. So to conclude this part, I want to point out that the net effect of within-host interactions, think about immunity versus resources, and the outcome is really going to depend on both the host and the parasite species and the exact mechanisms that are underlying those interaction effects. We're trying many other parasite combos in my lab too. Wolbachia seems to be really promising. Some of our more virulent co-infection or more virulent protozoa look really interesting. But I also want to point that co-infection could fundamentally change the outcome of host competitive dynamics. And this could have really important implications for thinking about the transmission of disease in populations, the persistence of disease in populations and in communities. And even, in an agricultural perspective, if one of these stored grain pests is a lot more damaging than another, these co-infections can really modulate any attempt to use biocontrol methods, for example, to control them. All right, and now I want to quickly go through this last part of my talk, where we're thinking about the evolutionary consequences of defending multiple fronts. So I want to point out that when people model inducible immunity, so when they're thinking about using theoretical methods to predict how inducible immune systems should evolve, there's a couple different assumptions that different modelers have made. So Shudo and Awasa originally said that, well, inducible immunity, let's say that, an individual gets infected at time T here, and then the immune system just turns on. So this is kind of a switch-like mechanism that's really just dependent on time post-infection. Frank and Hamilton, on the other hand, so Steve Frank used a combination of inducible immune parameters where parts of the immune response were only dependent on time. So if you get infected, you turn on your immune response. Other parts of the immune response, however, are dependent on microbe density. So it's not enough to be infected. That microbe has to be growing and feeding back as a positive feedback loop on the continued growth and dynamics of the immune system in order to perpetuate the inducible immune response. And in Hamilton, they only had microbe density. So basically, you're writing a differential equation where your immune response is completely dependent on the parasite density inside the host to continue amplifying. And you might ask, why, Anne, why are you telling me this? Why does this matter? This seems really picky. Well, the problem is that when we think about the cost structure of inducible immune responses, this really matters. So, you can model cost over the time course of infection from 0 to recovery, for example, or mortality here. And this is the cost of the parasite itself, right? So this parasite could be really virulent, in which case C is really high, or it could be pretty benign, in which case C is really low. We have the cost of the constitutive response here. So how much of the immune response you're making before you get infected, or you're maintaining during infection. And then you have the cost of the inducible immune response. And this is, depending on how much immunity you have over this time period. And so if you're turning on the immune response regardless of microbe density, this term is going to be entirely dependent on how long the infection lasts. Whereas if you're turning on in regard to microbe density, you're basically, this cost structure could be a lot lower because you're only paying the cost as needed, right? You're incrementally increasing your immune response. And so, if we can predict that if CP is relatively low, this kind of microbe density dependent term could be more important or provide better fitness to the host because you only pay the cost as you need to go. Whereas if this parasite is very virulent, this might ultimately be more beneficial because the time course that you need, the time it's going to take to kill the parasite is going to be lower. You're going to turn that immune response on maximally, kill all the parasite, and then recover faster. And so thinking about how we model these different parameters is important. The problem is there's not a lot of data, really, right? People will measure the inducible immune response, but they won't always measure how many parasites are in there and try to get the actual functional form of these different types of immune responses. So we try to do this. So we have Tribolium castaneum and Bacillus thuringiensis, and this is from some prior studies where we infected our larvae and we took a time course. And they do something like this. So the bacteria grows for a little while, then it levels off as the immune response kicks in, and then the host either loses and the bacteria wins and the bacterial load increases, or the host wins and the bacterial loses and the host manages to control its bacterial load. And so we can use this natural variation in these bifurcation dynamics to fit different models to the gene expression. So we can look at gene expression that's entirely dependent on time. So it doesn't matter what the bacteria are doing. It just matters how long since infection it's been. We can look at rapid inducible dynamics, so induction and decay dynamics. This might be really important for really pathological responses. And then we can look at things like immune responses that are correlated to bacterial load. And it turns out, so here example, we have the IMD and TOL pathways. When we map these different types of dynamics to these different pathways, you can see that a lot of the signaling pathways undergo these rapid inducible induction and decay dynamics, which makes sense because you want to tightly regulate these signaling pathways. But particularly for the IMD pathway, a lot of both the antimicrobial effectors and some of the recognition proteins that are under IMD control are all pretty tightly correlated to bacterial load. So they're receiving constant feedback from bacterial load in terms of their transcription. So given these myriad costs in multiple fronts, we should expect multiple or substantial natural variation in inducible immune parameters. And so we can, if we have number of microbes on this axis, for example, so we're looking at our microbe density dependent inducible responses, and we have the magnitude of the immune response. We could, for example, expect an increased baseline. So this would be analogous to that time dependent parameter. So no matter what your microbe load is, as soon as you get infected, you're turning on your immune response. And that's this parameter here. We can have an increased maximum. So this is your squirrel with a flamethrower. If you get infected enough, you're just, you're raising all the microbes regardless of the collateral damage that you might cause. You can have increased sensitivity. So it takes fewer microbes to turn on that immune response. It's microbe dependent, but it's going to take fewer microbes. Or you could have different slopes. And I think of this as twitchiness. So, as microbes increase, do you become that squirrel with a flamethrower faster? Or are you a little more circumspect? Are you waiting a little bit longer? You're a little bit, more nuanced in your ability to turn on your immune response. And so the relative slope could be a good explanation for, resistance later in infection, but it could also be an indicator of immunopathology or the potential for immunopathology. So we wanted to know how variable are the dynamics of inducible responses in wild populations. And so we went out to grain elevators and feed mills and things like that. We set these little pheromone-baited traps, and we catch a lot of beetles. I'm going to show you the results from 7 populations today. What we did is that we had 4 castaneum populations and 3 confusum populations caught from the wild. We infected them with 4 doses of either Bacillus thuringiensis or Photorhabdus luminescens. So they're both killer bacteria, but they have very different life histories. So we're thinking about the multiple front side of things now. And we sacrificed them at 8 hours post-infection. And the idea here was to get a range of bacterial densities at which we could compare antimicrobial peptide expression. So we're measuring survival, some of these pathway-specific inducible immune parameters, and resistance. And what we found, first of all, is that there is a lot of variation among our different strains and their ability to survive bacterial infection. So these are different populations. And then this is the dose response. And you can see that overall, I know it's kind of hard to see. There's a lot of dots on here, but you're just going to have to believe me. So if we look at GRT here, we have the lowest dose and then intermediate doses and the highest dose. And so as dose increases overall, bacterial load at 8 hours also increases. But you can see that most of these populations are fairly similar in here. And then we have this one population that is really resistant to these bacteria. And we have another busy plot here, but if you squint at one of these colors, for example, this pink line here, what we see is that this is an antimicrobial peptide, ataxin-1, and this is the relative bacterial density down here. And you can see that for most of these populations, there's a strong correlation between bacterial density and the expression of these antimicrobial peptides. So we do have a strong microbial density dependent term here. Now we have this one population, that's the same population that was really resistant, where we don't see this, right? So there's no, it doesn't seem like this particular antimicrobial peptide is being, there's no feedbacks from microbial density. They're just turning it on. But overall, both BT load, site, and their interaction, so the slopes, are significantly different among populations. What we find then is that when we actually look at the different immune parameters, I'm blazing through this because we're almost out of time, but and we look at the relative hazard ratios, so how these populations survive. We see a couple things come out of this data. So these circles are correlations that are significant. And we see that for Photorhabdus infections, the intercept, so as soon as you get infected, even if it's like with a sterile saline injection or something like that, as soon as you're wounded, how much do you turn your immune system on? And that's really predictive of Photorhabdus luminescens infection. So here we have the defensin 1 intercept, our antimicrobial peptide turn-on rate, and here we have the hazard ratio. So the more the stronger that an initial inducible response, regardless of microbe density, the more likely you are to survive if you're infected with Photorhabdus luminescens. For BT, on the other hand, there's no association with your that initial microbe-independent inducible response, but there is a correlation with that slope. So how twitchy you are, how quick you turn on your immune response in response to every microbe that you have in your body. But here, the effect is actually opposite. So the steeper your slope, the twitchier you are, the more likely you are to die. And so we can summarize this by saying, here we have, if you have an increased baseline, so regardless of the number of microbes you have, the faster and stronger you turn on your immune response, the more likely you are to survive against Photorhabdus luminescens. On the other hand, the twitchier your slope is, so the steeper that inducible response in regard to microbes, the worse you are in terms of your survival against Bacillus thuringiensis. And this is probably indicating immunopathology. So there's a lot going on in these immune responses. BT is a pretty virulent microbe, and it could just be that these steeper slopes are basically a panic response in the beetles. They're turning everything on really quickly, including things like burning through energy. So we know that IMD pathway, for example, is also modulating energy metabolism during an infection. So my hypothesis right now is that these individuals are just basically less tolerant of infection, and they have more immunopathology and more other kind of sepsis type effects in the infection. So to summarize my talk here, I just want to point out that there's not a universally optimal immune strategy. It depends on who you're infected with. Is it one parasite type or many? Are we talking about generalists or specialists in host communities? When you're infected, right? Is it early or late in life? Are you fully competent or are you immunocompromised? How you're infected. So how's the architecture of your immune networks influencing your ability to fight infection and your susceptibility? And then how often are you getting infected? Is it frequent or rare? And so I just want to point out that context-dependent evolutionary feedbacks seem to maintain substantial natural variation in immunity. And to give you kind of an idea of my broader vision here, the types of goals that we're working on in my lab, we basically want to understand this entire system, right? We want to think about how variation within different hosts, whether it be individuals of the same species but different life stages, or individuals within this community that are interacting either through energetic resources or sharing parasites, feeding epidemiological feedbacks onto each other. How does this variation within the host at the molecular level ultimately influence the ecology and evolution of these communities? And this isn't just relevant to beetles, right? We can think about grasslands, right? There are prairies out there that with many different grass species that share viruses, and you can think about, they have really interesting immunological dynamics. Or we can think about a system like Lyme disease, right? Where we have all these different hosts, whether they have variation across their life stages or variation among species that are feeding back into the disease system. So with that, I'll acknowledge my lab. People in bold did a lot of the work here. We're funded by various sources. I'd be happy to take questions. Thank you for listening to me.